Hi guys, in this video we're going to talk about the histology of the male reproductive system. We're going to look at the testes and we're going to see what happens to the sperms from the time they get formed in the testis itself, and we're going to see what happens to them and what happens to the epithelial lining as we move from the testes all the way through the retitestis, through the efferent ductules, and then to the epididymis, and there's the vas deferens, and then the two important glands that they form parts of the secretion, the semen itself, which are the seminal vesicle and the prostate. Now, if you guys look at the testis itself, you guys can see that. Remember that when we talked about the gross anatomy, I told you guys that we have an important layer of connective tissue that we call it the tunica albuginea. We're going to see that in histology. It's the very first layer that surrounds the epithelium and the structure, of the testis itself, all the way around it. And then remember that we had the visceral layer of tunica vaginalis, and we talked about that in the gross anatomy. So these are the things we can see as they surround the testis itself. And then remember that we said that we get once we get out of the testis, get into the retitestis, which is something at the back of the testis, all right? And then from there we get to something that takes the sperms away from the testis, so we call them the efferent ductules. It's all about a coiled tubules everywhere in the testis. And remember that when I told you guys in the previous video in the gross anatomy that the tunica albuginea, it sends septa that breaks down your tubules of the testis into around 200 to 300 seminiferous tubules, and then it's all again about tubules and ducts, and it's everywhere where you get to see your sperms formed in the testis getting transported through all these tubules all the way up until they reach the pelvic cavity. And then they get added up by the seminal fluid coming from the seminal vesicle and the prostate, and then they are ready to be ejaculated, right? So this is the general low magnification of a testis and what surrounds it, exactly like what you have in this diagram. And let's talk about what happens to the sperms, because if we understand what happens to the sperms and how do the sperms form, general talking, we're going to understand the histology of the seminiferous tubules and the seminiferous epithelium. So as you guys know that the sperms start off by the spermatogonia, and these are the diploid cells. They are 2N. They undergo mitosis, and from one cell they get from the spermatogonia A, they give you a B, which will continue the actual process that will take you all the way up until you reach the sperms, and it's gonna give you another spermatogonia A, which will go back and sit at the basement membrane to make sure that we can still have spermatogenesis forever, all right, or as much as we can. Okay, so spermatogonia A, it's gonna give you A and B. A will gonna sit back. It's gonna sit back to undergo the same idea again and again. And the first one will give you B, and B is gonna continue the process. Now from spermatogonia B, we're gonna have another mitosis, and that's gonna give me another two, yes, if it's of course two. It's gonna give me the diploid cells, which are the primary spermatocytes, and from here we're gonna undergo meiosis. All right, 1 and 2. 1 will give you the secondary spermatocytes, and 2 will give you the round spermatids. And then once we reach the spermatids, now we are ready to do a morphological change to the spermatids from round to late to the spermatozoa, and this process needs the testosterone that comes from the surrounding interstitial cells, the Leydig cells. All right, so again, remember that what happens here is spermatogenesis, okay, and that means mitosis and meiosis. However, the spermiogenesis the last thing that will give you the final shape of spermatozoa. It's basically a morphological change to what we have reached at this point. It is not mitosis, it's not meiosis, it's not differentiation and um, proliferation. It's only morphological change to the final results that we get here. So remember that very important information, and that is going to be done under the influence of the testosterone. We're going to talk about that as we proceed. All right, so this is what happens to the cells briefly. Okay, now, if we look at this, we have the seminiferous tubule. This is the epithelium. Let's suppose that this is a seminiferous tubule, a huge tubule. This is my lumen. So all of these are the stratified seminiferous epithelium. We're going to have the spermatogonium at the basement membrane. We're going to proceed all the way until we reach the lumen. So as we proceed, we're going to have the mitosis, the meiosis, and then at the final stage here, we're going to have the morphological change to get the sperms. Remember that we have a very important cell called Sertoli cell, and that cell is it looks like oval shaped. You can see it looks like triangular shape with elongated nucleus, whatever you want to call it. It's a very important cell. All right, this cell is very important because it forms the blood-testis barrier. It's actually as you look at the neighboring Sertoli cells, you guys can see tight junctions, right, between um, the lateral surfaces of the Sertoli cells all around the testis, and this is what's called the blood-testis barrier. You get to see that tight junction after the spermatogonia, because spermatogonia is 2N, and they have the genetic material identical to everything around them, so there is no harm that we have them facing the blood capillaries. The problem is for the differentiating cells here, we're going to get to see genetic material that is different than the surrounding blood cells. So thanks to the Sertoli cells that are going to form the blood-testis barrier, that is going to make sure to kind of separate and protect the developing spermatocytes with the different genetic material from the circulating blood and macrophages and immune cells, because if they come to know that we have a different kind of cells, they're going to attack them. All right, so this is one importance of the Sertoli cells, as they form the blood-testis barrier. All right, and they're gonna make sure to let only the nutrients, the oxygenations, or whatever we need from the blood to get into the seminiferous tubules and get to the cells without letting these immune cells to enter the tubule itself. Another thing about the Sertoli cells, they are important because they are giving the nutrition, the support, other than the blood-testis barrier, to all the growing spermatocytes. One important thing also about the Sertoli cells is the idea that they secrete the androgen binding proteins, which are very important because these proteins are going to catch the testosterone, the androgens secreted by the interstitial cells or the Leydig cells, and they're going to catch them by the androgen binding proteins. They're going to bind themselves into them, bringing them all the way to the seminiferous tubule, and that will help at the final stage, which is the spermiogenesis, which is the morphological changes of the spermatids, so that they can transform into the final look of the sperms. So this is very important function of the Sertoli cells. So they're gonna catch the testosterone, bringing it all the way to the luminal side, where we can have our spermatids developed and undergo spermiogenesis to form the sperms or the spermatozoa. All right, so these are the important functions of Sertoli cells. So other than that, we talked about the cells, all of the cells of the seminiferous tubule, talked about the Sertoli, we talked about the interstitial or Leydig cells, and let's look at how would this look like under the microscope. So this is a nice diagram showing how things would look like. So if you guys look at the very basal part, you get to see the spermatogonia. So if you have any cells touching your basement membrane, that is going to be the spermatogonia. Now, if you look at a cell that gives you the string-like appearance, it's kind of as if you're trying to do these lines. All right, if you guys look at this one and that one, okay, stringy appearance of the chromatin of the cell, this is going to be your primary spermatocytes. All right, so if you have cell that is touching the basement membrane, that's going to be the spermatogonia. If you have a cell that gives you the stringy appearance of the chromatin and of the nucleus, that's going to be your primary spermatocytes. Okay, so and then as you proceed to the luminal side and you get to see those this is actually your sperms or spermatozoa. Okay, if you guys see a circle that looks like an ink spot, this is actually your spermatids. Okay, so I hope you guys now have an idea on how to identify the differences between the spermatogonia. If you have a cell that again looks like an ink spot that is touching the basement membrane, this is your spermatogonia. If you have any cell with a stringy appearance, okay, this is going to be your primary spermatocytes. All right, primary spermatocytes. The secondary spermatocytes, we cannot see them, we cannot catch them. They very quickly change into the spermatids. All right, so you don't have them. Okay, and if you see something like ink spots, okay, it's not giving you those stringy appearance of the chromatin of the nuclei. Okay, this is actually your spermatids. Okay, if they are ink-like spots, spermatids, because here we are not at the basement membrane. If we were at the basement membrane, it would have been the spermatogonia. Okay, but it's not, because here we are in the center, somehow near to the lumen. Okay, now once you have these, this is actually the look of the sperms. Okay, the spermatozoa. So if you guys look at this is actually spermatozoa, or sometimes we call it late spermatids, but that's fine. You guys can say spermatozoa or sperms. Okay, now if you look at these cells, which have an oval shaped cells, can you guys see these oval shaped cells? These are actually your Sertoli cells. I made a square around them. Okay, so basically this is your Sertoli cells, you guys. Okay, so all of these are your Sertoli cells. Okay, so I hope now it's easy for you to identify different types of the cells in the seminiferous tubules. So here we have another nice picture showing you guys that if you have cells touching the basement membrane, that would be spermatogonia. If you look at cells that gives you kind of these so many lines inside, dots, stringy appearance. Okay, so this is going to be the primary spermatocytes. If you have the oval shaped or triangular shaped kind of cell, this is your Sertoli cell. If you have kind of a circular cell that looks like ink spots, this is actually your spermatids. And then these are your late spermatids or spermatozoa. Okay, so here we can say late to make it easy, or we can also say spermatozoa. All right, this is at the luminal side. Now, when we proceed, we get to see how the retitestis is actually looking like. It's basically a coiled ductules here. you have all these spaces for the sperms to get propelled from the seminiferous tubules. Once you get to see all of these kind of epithelium, tubuloalveolar structure, and you have your lining epithelium, and with all of these spaces to get your sperms away from the seminiferous tubules, and you can see seminiferous tubules, it means here you are at the retitestis. Okay, so once you get to see a seminiferous tubule, and what's neighboring to that is mucosal folds of a tubule with a core of connective tissue, and the lining epithelium here is actually somehow simple columnar epithelium. It cannot really be seen because it's low magnification. But once you see that you have mucosal folds that is near to the seminiferous tubules, you can say definitely that this is the retitestis. Okay. And as we proceed and we have a cross section of the epididymis, what's very, very famous about the epididymis is that they have the pseudostratified columnar epithelium with stereocilia. With what? With stereocilia. It's an apical specialization of the epithelial cells. I have here a columnar cell. So what happens at the apical surface, I get to see very long kind of elevated structure. Can you guys see how the stereocilia are very long? And here, although they are called stereocilia, but they don't act like the cilia, they don't move. The function of this apical specialization is to basically absorb as much fluid as possible, because we needed that fluid. Because when we were in the testis, it was very important to have this fluid to help the sperms to move all the way from the testis to the epididymis. But because at the epididymis, the sperms are going to learn how to swim, they're going to mature enough. And at that point, we don't need all of that fluid as we get out of the epididymis all the way through the vas deferens to get into the next destination. Here we already know how to swim, so we don't need that much of fluid. So thanks to the stereocilia, it's gonna absorb a lot of fluid, okay, and get the sperms matured, stored, and then they're gonna get moved and propelled to the next station through the ductus deferens, and then to get into the pelvic cavity. Okay, so this is how the epididymis would look like. Now, as we proceed, and now we are away from the testis, we're away from the maturation factory, and now we are ready to get all the way through our bus, which is the ductus deferens. And this is a muscular tube, which is very important tube, because you guys know that it brings the sperms all the way from the scrotum side, bringing it from outside, from the perineal region, all the way up, and it's gonna move it by paracelsus movement, thanks to the 3 layers of smooth muscles that we have them in the ductus deferens, and that what makes the ductus deferens unique. And the distinctive feature of it is the 3 layers: the longitudinal, inner and outer layers, and middle circular layer. A lot of smooth muscle layers that would help by contraction of them to bring the sperms all the way from the epididymis all the way through the inguinal canal inside to the pelvis, to bring them to the ampulla, and then to the ejaculatory ducts. So this is how we would know how to identify the ductus deferens. And then if you look at the lining epithelium, it's going to be again the pseudostratified columnar epithelium, or sometimes shorter kinds of cells. But again, we are in the same concept here, that we need to have these very busy cells with the pseudostratified, and again, the muscular layers outside are going to help you to identify the histological section of the ductus deferens. All right, and of course, after you have your epithelium, your connective tissue, because you guys know that by definition the epithelial lining cells, they don't have, they are avascular, they are lacking a lot of important features. So thanks to the next layer, which is the lamina propria, which is the connective tissue that's going to take care of the nutrition, the lymphatics, the innervation, anything that would be needed in the epithelial, it's going to be ensured by the next connective tissue layer that comes after the epithelium, which is the lamina propria. And then we have the muscular layers, and then at the very outermost one, which is going to be the adventitia, we're going to have another connective tissue, which is having the blood vessels and everything else that we would need. And that would be in the last layer of all of the layers of the ductus differens or vas differens. Now, moving on to the first gland that we have in the male system, which is a very important gland, it's the seminal vesicle. Now, again, we have muscular layers because, again, we need to have contraction of these muscles to help us to get the secretions out of the glandular tissue that we have here. We have epithelial lining, and the epithelial lining of seminal vesicle would be a simple cuboidal cells, and it's a glandular tissue. And if you have highly folded mucosal lining, so if you guys look at this, it looks like so many folds of epithelium, and they are forming kind of network, and you have your lumen everywhere around you, because once you secrete the fluid, it needs to get into the lumen, and then it needs to, and this is where you have your seminal vesicles, because we brought the sperms all the way through the ductus deferens, and from here they're going to get into the ejaculatory duct, and the seminal vesicle is going to secrete its secretions into the ejaculatory duct and bringing them to the prostatic sinuses. And from the prostatic sinus, we're going to add up the prostatic fluid, and the ejaculatory ducts are going to open into the prostatic urethra, and then all of them will going to be forming the 100% content of the semen. We have 70% coming from the seminal vesicles, like we said in the previous video. We have like 20% prostatic secretions and like 10% the sperms themselves. Now, if you guys look at the highly folded mucosal lining of seminal vesicle, you will get to know that this is definitely the seminal vesicle because it has a very highly folded epithelium. The purple thing is actually your cuboidal epithelium. All right, the pink thing is your lamina propria, because again, yes, we have epithelial lining, but we need the connective tissue that supports that and takes care of everything that the epithelial cells would ever need. And this is what we call the lamina propria. So basically the core is going to be your lamina propria. It's nothing but the connective tissue that is responsible for everything that your epithelial cells would ever need. And once we pass that pink color, we are coming here into smooth muscles that they have nuclei, the inner circular and outer longitudinal. And then once you get to see blood vessels, venules, and arterioles, you know that you are again in the connective tissue that wraps around the whole thing and that takes care of the outermost layers. And this is your adventitia. All right. Now, we know that the seminal vesicle is a gland, and also the prostate is a gland. So how can we differentiate between them? If you guys look at the prostate, remember that when we talked about the gross anatomy of the prostate, we said that we have, first of all, we have something called the fibromuscular stroma. The anterior loop is basically formed of fibromuscular tissue. So you get to see a lot of smooth muscles everywhere. And so you get to see a fibromuscular stroma around you, okay? It's not highly folded epithelium. It is folded, but it's not as much highly folded as the seminal vesicles. And if you look at the gland itself and you look at the next layer, you will get to see your fibromuscular stroma is everywhere. So what's different, guys, here from there is here you have the folded epithelium. It's just your epithelial lining, and that pink is actually the lamina propria. Okay, and the whole thing here is actually your lumen. So what happens here is gonna continue all the way here, and that is all your lumen. But if you look at the prostate, you're gonna see that there is the pink and dots everywhere filling in between the cells. So it means that it's not highly folded. Yes, we have folds of mucosa, but it's not as much folded as here. Okay, so here you have cores of fibromuscular stroma, you have the smooth muscles. Here we don't have smooth muscles in the core. It's actually, this is only the mucosal fold, and you guys know that the mucosa means I have the epithelial lining and the connective tissue that supports it. It's only fibroblasts and fibers, collagen fibers, and the uh, um, the uh, uh, the hyaluronic acid or the extracellular matrix that we have in connective tissue. Okay, so this is basically mucosal folds. But if you look at this, you guys can see a lot of smooth muscles in between. It means these are not folded mucosa. It's just this is the only fold we have here. And if you guys look at this, you don't see it highly folded as there. Another thing that will make your life very easy in the prostate, and it's gonna give you a very important clue, is the presence of the prostatic concretions. All right, so these prostatic concretions are kind of spots, looks like ink spots, but light in color. And these are actually some kind of um, um, prostatic secretions that would stay there and increase with age. So you get to see them in the lumen of your prostatic gland. Okay, so whenever you see this, all right, and you see a glandular tissue, fibromuscular stroma, you are 100% sure that this is a prostate. Okay, it looks way different than the seminal vesicles. I hope you guys now you know how to differentiate between different parts of the male system. Thank you guys for watching. Don't forget to like and subscribe. I mean, subscribe, and see you in the